Welcome to this deep dive into Radon's Smart Review RadBio. Today we're focusing on how tumors build their lifeline angiogenesis and the master switch controlling it all, HIF-1 alpha.
Exactly. We've talked before about tumor hypoxia, low oxygen, and how it affects radiation response.
Right.
So today, we'll really drill down into how those hypoxic conditions actually, drive the formation of new blood vessels. And HIF-1 alpha is right at the heart of that. Super fundamental stuff for radon-lick.
Okay, let's tackle angiogenesis first then. What is it really?
Well, think of an established tree, with branches reaching out.
Okay.
Angiogenesis is like that tree sprouting new branches to get resources where they're needed. It's new blood vessels forming from ones that already exist.
Got it. So sprouting, not building from scratch.
Precisely. That building from scratch idea, that's more like vasculogenesis, de novo vessel creation from precursor cells, like building a whole new road network. But for tumors, it's overwhelmingly angiogenesis, the branching out, that gives them their vascular network.
Makes sense. And they need this because, well, a tumor can only get so big just by diffusion, right? Maybe a millimeter or two.
Exactly. Beyond that tiny size, diffusion of oxygen and nutrients just doesn't cut it. It's like trying to power a city with, a single extension cord.
Yeah. Not going to work.
So it absolutely needs its own dedicated supply lines. And this is where the angiogenic switch comes in.
The angiogenic switch, I like that term.
Yeah, it visualizes it well. It's that moment, the balance tips. You've got pro-angiogenic factors saying grow, build vessels.
The accelerators.
Right, and then you have anti-angiogenic factors, the stop signals, the brakes. In healthy tissue, those brakes are usually on. But tumors, they're masters at manipulating things to flip that switch to on. They're basically shouting, we need more blood supply now.
So let's meet some of these key players then. Maybe start with the pro-angiogenic team, the accelerators.
Okay, top of the list, the big one is VEGF, vascular endothelial growth factor.
VEGF. Heard of that one.
Yeah, definitely the most well-known, probably the most potent promoter. Think of VEGF as the foreman on the blood vessel construction site.
Okay.
It's actually a family, VEGF-A, B, C. But VEGF basically tells endothelial cells, the vessel lining cells, multiply, move, form tubes.
And who's telling VEGF to do this? Often HIF-1 alpha, right, which we'll get to.
Exactly. HIF-1 alpha is a major driver. Then there's BFGF, basic fibroblast growth factor, also called FGF2.
What's its role?
Think of BFGF as maybe the supportive engineer. It encourages endothelial growth, but also helps them resist apoptosis-programmed cell death, often works alongside VEGF.
Gotcha, teamwork.
Then you have PDGF-BB, platelet-derived growth factor BB.
Okay, another factor.
Yeah, this one's more like the quality control manager. It's crucial for recruiting parasites.
PARAB, ECs.
Right, those cells that wrap around new vessels. And smooth muscle cells, too. They help stabilize the new structures. VEGF builds the road. PDGF-BB helps make it durable.
Nice analogy. And wasn't there one more? IL-8.
Yes, interleukin-8, IL-8. This one's kind of a double agent. It promotes angiogenesis, yes, but it's also linked to increased tumor invasion and metastasis. So it helps the tumor get bigger and helps it spread.
Okay, so those are the accelerators. What about the brakes, the anti-angiogenic factors?
Right. The other side of the balance. Healthy tissues produce these to keep everything in check.
Examples?
Well, there are thrombospondin 1 and 2. Think of those as well-known natural inhibitors, just sort of baseline brakes.
Okay.
Then you have angiostatin and endostatin. These are really interesting.
Why is that?
Because they're actually fragments, like little pieces broken off from larger proteins, plasminogen for angiostatin, collagen 18 for endostatin. But these fragments themselves are potent anti-angiogenic factors.
Wow. Repurposed parts.
Exactly. Endostatin, for instance, can make endothelial cells undergo apoptosis and stop them from moving. Pretty powerful stuff.
Any others?
Yeah, interferon alpha and beta also have anti-angiogenic properties. And even heparin, which we usually think of as a blood thinner, can inhibit angiogenesis in some contexts. It's complex.
It really is. So the angiogenic switch flips when the pro-factors like VEGF and BFGF overwhelm these anti-factors, like thrombospondin and endostatin.
That's the core idea, yes. The balance tips and the tumor starts building its network. But, and this is important, the angiogenesis driven by tumors is often really chaotic.
Right, I remember reading that. The vessels are leaky, tortuous.
Exactly. Leaky, twisted, poorly organized. It's a flawed construction project, which paradoxically can mean some areas still end up hypoxic.
So building vessels doesn't always fix the oxygen problem.
Not perfectly. And worse, those leaky, disorganized vessels can actually provide escape routes for cancer cells to metastasize.
So it's an imperfect system, but good enough for the tumor to grow and spread.
Sadly, yes. It works for the tumor's purposes.
Okay, so that's the what and why of tumor blood vessels. Now let's get into the how, the master regulator you mentioned, HIF-1 alpha. You said this is big for board.
Oh yeah, HIF-1 alpha, hypoxia-inducible factor 1 alpha. This comes up a lot. It's a transcription factor.
Meaning it controls genes, turns them on or off?
Precisely. It binds to DNA and dictates gene activity. Now, HIF-1 itself is actually a dimer, 2 parts joined together.
Okay.
There's HIF-1 alpha, the star of our show today, and HIF-1 beta. HIF-1 beta also has another name, RNT. That stands for aryl hydrocarbon receptor nuclear translocator. Bit of a mouthful.
RNT. Got it. So alpha and beta, what's the key difference?
The key is regulation. HIF-1 beta, or RNT, is pretty much always there in the cell. It's constitutively expressed.
Always on.
Right. But HIF-1 alpha is the regulated part. It's the component that actually senses and responds to oxygen levels. Think of HIF-1 beta as the lamp that's plugged in, but HIF-1 alpha is the bulb that only lights up when it's dark, or rather when oxygen is low.
Clever analogy. So the cell has ways to sense oxygen and control this HIF-1 alpha bulb. How does that work when there's plenty of oxygen? Normoxia.
Okay, normoxic conditions, plenty of O2. Here, a family of enzymes called prolyl hydroxylase domain enzymes. PHDs are the HUOs. You might also see HPHs.
THDs.
Yeah.
Oxygen sensors.
Exactly. They function as cellular oxygen sensors. And what they do is they use molecular oxygen.
Need the O2.
Absolutely need it. To hydroxylate HIF-1 alpha. Specifically, they add an OH group, hydroxyl group, to certain proline residues on the HIF-1 alpha protein. It's like sticking a little chemical tag on it.
A tag that only gets put on if there's oxygen available?
You got it. And this hydroxyl tag is like a red flag. It gets recognized by another crucial protein, VHL, the von Hippel-Lindau tumor suppressor protein.
VHL. Okay, so PHD uses oxygen to tag HIF-1 alpha. VHL sees the tag. What then?
VHL acts like a molecular handcuff. It binds to the tagged HIF-1 alpha, and then it recruits other proteins that attach another kind of tag ubiquitin molecule.
Ubiquitin. That's the tag for destruction.
Right, that's the one. It's like a molecular kick-me sign. Marks the protein for disposal.
So HIF-1 alpha gets tagged with hydroxyl groups by PHDs, then VHL binds, leading to ubiquitin tagging.
And once it's covered in ubiquitin, the proteasome takes over.
The cell's protein shredder.
Exactly. The proteasome recognizes that ubiquitin signal and just degrades the HIF-1 alpha protein. Gone.
So under normal oxygen, HIF-1 alpha is constantly being made, tagged, and destroyed. Its levels stay low.
Precisely. Oxygen basically keeps it on a very short leash via the PHD-VHL proteasome pathway.
Was that the only control?
Not quite. There's another layer, an enzyme called FIH1 factor inhibiting HIF-1 alpha.
The FIH1.
FIH1 can also hydroxylate HIF-1 alpha, but at a different spot, an asparagine residue. This modification acts more like a dimmer switch.
How so?
It prevents HIF-1 alpha from properly interacting with essential coactivator proteins like P300 and CBP, which are needed for it to fully turn on gene transcription.
Ah, so even if some HIF-1 alpha escapes degradation, FIH1 can still dampen its activity if there's enough oxygen.
Exactly. So, normoxia recap. Oxygen fuels PHDs and FIH1. HIF-1 alpha gets hydroxylated. This leads to VHL binding and proteasomal destruction, and blocks interaction with coactivators. Net result, very little active HIF-1 alpha. Oxygen keeps it down.
Okay, crystal clear. Now the critical part.
Yeah.
What happens when things get hypoxic? Low oxygen.
Right, this is where HIF-1 alpha gets its moment. When oxygen levels drop, the PHDs can't work properly. Remember, they need oxygen for that hydroxylation reaction.
Power goes out for the gatekeeper.
Good way to put it. No hydroxylation means no tag.
And if there's no tag, VHL can't bind.
Exactly. The security guard doesn't see the red flags.
So HIF-1 alpha doesn't get ubiquitinated.
Correct. It escapes degradation, it becomes stabilized and starts to accumulate inside the cell. The kick-me signs aren't getting attached anymore. Some evidence suggests phosphorylation might also help stabilize it here.
So it builds up. Then what?
Then, this stable HIF-1 alpha translocates it, moves into the cell nucleus, where the DNA is.
The control center.
Right. And once inside the nucleus, it finds its partner, HIF-1 beta, remember RNT.
The one that's always there.
Yep. They join forces, they dimerize, forming the active HIF-1 transcription factor complex. The bulb connects with the power supply.
And this active HIF-1 dimer, Now it can bind to DNA.
Yes. It seeks out specific DNA sequences called hypoxia response elements, HREs.
HIE.
Because HREs are like specific addresses located in the promoter regions, the control switches for a whole bunch of target genes. And remember, HIF binds HREs. Don't mix it up with HSF, heat shock factor, which binds heat shock elements. Different pathway.
Got it. HIF for hypoxia, so it binds the HREs.
And because FIH1 activity is also reduced when oxygen is low, the HIF1 complex can now effectively recruit those co-activators we mentioned, P300 and CBP. The dimmer switches off.
Full power to gene activation.
Exactly. So under hypoxia, the whole situation flips. HIF1 alpha is stable, active in the nucleus, bound to HREs, and it powerfully drives the transcription of genes that help the cell survive and adapt to low oxygen.
And top of that list of genes is VEGF.
You got it. Hypoxia leads to HIF1 stabilization, which cranks up VEGF production, which drives angiogenesis. The tumor tries to fix its own oxygen problem. A classic feedback loop, really.
A very effective, if problematic, loop. But you said VEGF is just one target. There are others.
Oh, yeah. HIF1 controls a whole suite of genes, maybe 50, 60, even more, all geared towards hypoxic survival and often, unfortunately, tumor progression.
Like what else?
Well, besides VEGF for angiogenesis, it hits genes involving glycolysis, metabolism.
Ah, the Warburg effect connection.
Exactly. It upregulates GLUT1, a glucose transporter, to pull more sugar into the cell.
For fuel.
Right. And it activates enzymes like LDHA, lactate dehydrogenase A, and PDK1, pyruvate dehydrogenase kinase 1. These basically shift metabolism towards anaerobic glycolysis, making ATP without oxygen, though less efficiently.
So it helps the cell make energy even when oxygen is scarce.
Precisely. It also targets genes for erythropoiesis, making red blood cells, but that's probably more systemic. And importantly for us, HIF1 also activates genes involved in tissue remodeling and metastasis.
Making hypoxic tumors more aggressive and invasive.
That's often the case, yes. Hypoxia isn't just about survival, it can promote spreading.
Any others worth mentioning?
CA9, carbonic anhydrase 9. That's involved in regulating pH in the often acidic tumor environment. Another HIF target.
Wow. So HIF1 is like a central command hub for the hypoxia response? Angiogenesis, metabolism, survival, invasion.
Absolutely. It coordinates so much. That's why understanding it is just critical in radiation oncology.
Totally see that now. And the clinical relevance seems huge.
It really is. I mean, first, tumor growth itself, beyond those first couple of millimeters, depends on angiogenesis. And metastasis is often driven by hypoxia and HIF1 activation, leading to that more aggressive behavior.
And treatment resistance.
Big time. We know hypoxic cells are resistant to radiation, right? And some chemos, too. HIF1 activation is a key reason why it turns on survival pathways that protect the tumor cells from treatment stress, like activating a cellular survival kit.
Which naturally leads to thinking about targeting this pathway therapeutically.
Exactly. A major approach has been anti-angiogenic therapy. Drugs like bevacizumab Avastin, which is an antibody against VEGF.
Trying to block the vessel growth, starve the tumor.
That's the idea. There's also this concept of tumor vascular normalization.
Normalization, how does that work?
The theory is that maybe these drugs initially prune the really chaotic, leaky vessels, leaving behind slightly more functional ones for a while. This could potentially temporarily improve oxygenation and drug delivery.
Ah, temporary window.
Maybe. But it's complex. Sustained therapy can sometimes worsen hypoxia. The results with these agents, especially combined with radiation, have been, well, mixed. It's still an active area of research, figuring out the best way to use them.
And what about hitting HIF1 directly?
That's another major strategy being explored. Developing HIF1 inhibitors, drugs that block its activity or stop it from becoming stately in the first place. Lots of compounds being tested, things like topotecan, 17AG, 2Me2, have shown some activity.
Makes sense to go after the master switch itself. Okay, maybe we can distill some key takeaways. Clinical pearls.
Good idea. Clinical pearl number 1. Tumors need angiogenesis to grow beyond just a tiny size. This process is triggered by the angiogenic switch, basically when pro-angiogenic factors outweigh the anti-angiogenic ones.
Pearl number 2. HIF1 alpha. It's the master regulator for hypoxia. Low oxygen stabilizes it, letting it turn on genes like VEGF for angiogenesis, but also genes for metabolic adaptation, helping the tumor survive. Remember, low O2 by HIF on by GGF, up vessels grow.
Nicely put. And clinical pearl number 3, this whole balance between pro and anti-angiogenic factors, which is heavily influenced by HIF-1 under hypoxic conditions, is a key vulnerability in cancer. It's a critical target for therapy.
Excellent pearls. Ready for a quick board review, please?
Let's do it.
Okay, question 1. Which of the following is the primary pro-angiogenic factor directly stimulated by HIF-1 alpha in response to hypoxia? Is it A, thrombospondin 1, B, endostatin, C, VEGF, or D, interferon alpha?
Primary factor stimulated by HIF-1. That's got to be C, VEGF. The others are mostly inhibitors, except maybe interferon, which is complex, but VEGF is the main direct target for angiogenesis driven by HIF.
Correct. C, VEGF. Rationale. HIF-1 alpha transcriptionally activates VEGF, the key driver. Thrombospondin and endostatin are inhibitors.
Okay, next.
Question 2. Under normal oxygen conditions, how is HIF-1 alpha activity primarily regulated? A, increased dimerization with HIF-1 beta. B, enhanced binding to HREs. C, hydroxylation by PHGs leading to VHL-mediated degradation. Or D, direct activation of VEGF transcription.
Normal oxygen, primary regulation, that's the destruction pathway. So C, hydroxylation by PHDs allows VHL binding, ubiquitination, then degradation.
Correct again. C it is. In normoxia, PHDs tag HIF-1 alpha using oxygen. VHL binds the tag leading to destruction. A, B, and D describe what happens under hypoxia.
Good differentiation.
Question 3. Which of these is a well-known endogenous inhibitor of angiogenesis? A, BFGF, B, VEGF, C, IL-8, or D, endostatin?
Endogenous inhibitor. Well, A, B, and C are all pro-angiogenic, we discussed, so it must be D, endostatin, that fragment of collagen X8.
Exactly. D, endostatin. Potent natural inhibitor.
All right, last one.
Final question. What is the key transcription factor complex responsible for sensing hypoxia and initiating the angiogenic response?
Yeah.
A, NFKB, B, AP1, C, HIF-1, the HIF-1 alpha HIF-1 beta dimer, or D, P53?
Key complex for hypoxia sensing and angiogenesis. That's the main player we've been discussing. C, the HIF-1 dimer, formed when HIF-1 alpha stabilizes and joined HIF-1 beta, or RNT.
Perfect. C, HIF-1, it binds to the HREs of genes like VEGF. And crucially, it's HIF binding HREs, not HSF binding HSEs.
Right, I gotta keep it straight.
Okay, great, Blitz. So, just to quickly wrap up our deep dive today, we've really unpacked angiogenesis, how essential it is for tumors getting bigger, and how it's controlled by that angiogenic switch, the balance of pro and anti-signals.
And we spent a lot of time on HIF-1 alpha, the absolute linchpin sensing low oxygen. Under normal oxygen, destroyed. Under hypoxia, it stabilizes, heads to the nucleus, partners up with HIF-1 beta,
and turns on a whole bunch of genes, most famously VEGF, to drive that new blood vessel formation, plus all those metabolic adaptations, too.
Yeah, and we saw the clear clinical relevance, how this pathway fuels tumor growth, metastasis, resistance to our treatments, and why it's such an important therapeutic target.
Absolutely essential concepts for anyone in our field. Understanding hypoxia, HIF-1, and angiogenesis is just fundamental.
Couldn't agree more. It really forms a cornerstone of modern cancer biology and treatment strategy.
Well, you can complete practice oral boards over at radocsmartlearn.com and definitely subscribe to Radonc Smart Review for our next RadBio deep dive. We'll be tackling cell cycle kinetics next time. Thanks for tuning in.